So welcome, Dr. Baccarelli, Professor of Genetics and Molecular Medicine. We will be sharing this session here, looking at pathways, the genetics of suicide. You know, a lot have been said about suicide, but you know, as a one who teaches molecular medicine, you know that there is a molecular mechanism which is really the origin of all diseases. And so I think we're about today to share this session to bring that, to give us some insights as to what are the metabolic pathways, the molecular pathways that are involved in suicide. And so I'll give way to Professor Andrea Baccarelli to go first, and then I will go after. Over to you, sir.
Thank you so much for the invitation to be here today, for the very kind introduction. And I'll get started by bringing up my screen so you can see my slides over here. And here we go. So after thinking about what I could have presented today, I decided to focus my presentation on a specific subset of molecules, which are the epigenetic pathways, because most of my work and my expertise is in doing this type of epigenetic studies. And I'll try to start by motivating why you should care about epigenetics. So I'll first want to give you a standard introduction. So epigenetics is about programming or gene expression that does not depend on the DNA, and number two is stable. And I mean, this is an important consideration because you want, this is about marks that are different from the DNA sequence, but they still control gene expression. But there is almost everything in the cell that controls gene expression. And so to be specific, epigenetics is about those regulators of gene expression that can be stable through cell division, so through cell mitosis or meiosis. And there is this interesting idea of transgenerational inheritance that we will touch upon at the end of my presentation. For the purposes of my presentation, I want to highlight two characteristics of epigenetic programming. First of all, epigenetics is modifiable. And that is very different from the genome. The genome is not modifiable, or at least changes very little. And also, epigenetics is about the genes that are active or poised to be activated. So there are different states of genes. So it is particularly important because we are really interested in understanding genes that are active now, but we are even more interested at understanding what are the genes that are ready to be activated. And this is where the word programming comes about. It's a word that in biology we tend to make complicated, but in reality it's not different from our programs or our plans for the weekend. I mean, all of us have made plans for the weekend or for the holidays. And we already have plans. I mean, at some point we will act upon them. Perhaps we already started to think about gifts and things like that. But we can still change, correct? We can still change them. It is really particularly important because potentially the epigenome is associated with current health, but also with the future health. So here I would like to specifically give you the same type of definition translated into an example. And this example is a musical example. And so what you see here on the left-hand side is the DNA. And this is particularly important for us because the DNA here is a book of music. It's printed, and you can think how someone who has good musical skills can do a lot with this book, and they can get you a phenotype, which is actually the actual symphony you can attend to and enjoy. And you can understand also how from the same DNA, you can have entirely different symphonies. You can change the orchestra, change the conductor, change the music hall, the environment where the symphony is taking place. And you can really have very, very different phenotypes. And this is something that in genetics is very well known. The genome is important, but there is a lot of variation that in the way the genome is expressed, is activated. And you can have people with identical genomes, one of them getting disease and the other doesn't, and so on. So this is certainly important to keep in mind, but this doesn't tell us yet where the epigenome is. And in fact, if you wanted to see the epigenome, you wanted to go on stage, step onto the stage, and look over the shoulders of each of the performers. And if you were allowed to do so, you will see something very different. You will see that something like that, perhaps. You will see that the sequence of the nodes is exactly the same, but there are marks added onto the nodes that do not change the sequence, but they do change how the sequence is activated, expressed, and read. And this is what happens with the epigenome. The epigenome is about markings added onto our DNA that does not change the DNA sequence, but it changes the way the sequence is activated, particularly when and how. And this is particularly important because all of our cells have 20,000 genes. All of our life, we keep carrying our 20,000 genes. So they're exactly the same across our body. They're exactly the same across our lifetime. But in each of our cell at any given time, we only have 10% of those 20,000 genes that are active. And this changes over time, changes between cell types and tissues and between individual cells. And so we have this miracle of biological engineering that our cells do for us, where they exactly control and decide which genes are on and which genes are off. And this achieved mostly through epigenetics programming. At least epigenetics programming is one of the layers that contribute to these decisions. So at this point, I think I can step out of the analogy. And actually, no, I can finish the analogy because I want to also make a point that in epigenetics, you can see different types of programming. And here you can see how this musical sequence is particularly changed. And there are places that are completely inactivated, suppressed. In molecular biology, we would say silenced. But what is really interesting for my presentation is that we see on this page markings that are in ink, which once they're applied, they never change, and markings that are in pencil that once applied, they can be erased. Perhaps erased also very quickly. And we have example of these dynamics in the epigenome. For instance, we have epigenetic marks that determine whether during embryogenesis, what type of cells each of the cells in the embryo become. And for instance, there will be cells that become brain cells, cells that become liver cells. And the liver cells will express liver genes. The brain cell or the neurons will express neuron genes. And they will not express the other cell types genes. This type of decisions are driven by epigenetics, and they never change. It's very hard for a neuron to decide to stop producing neuron genes. However, there are some in pencil. For instance, the epigenetic machinery that controls immune genes, cytokines. those can go up and down in seconds, and there is evidence that in a few minutes, the epigenome of those genes can change completely. And there is all a world, a range in between, of course. So you can think about a continuum of these genes that are regulated over different timelines. So the type of epigenetic mechanism I'm going to review with you today is DNA methylation. DNA methylation is a very simple chemical modification of the DNA. It's a methyl group, CH3, which is added onto the DNA. It's a chemical modification. It's bound to the DNA with a covalent bond. And methyl groups can activate or most commonly silence genes. And so this is where we get, and for our purposes, we can think that when a gene is methylated, that gene is silenced, inactive. Okay, so I would like to cover over the 30 minutes that I have left, 3 points here, 3 parts. One, I would like to show you how psychosocial stress affects the epigenome and why that is important using evidence from animal models. Then in our lab, we do human studies, and I'm going to show you a couple of examples, at least, of our human studies. And finally, I would like to cover a part that is particularly interesting as to whether changes in the epigenome that happens now can be passed on to our children, our grandchildren, and in general to future generations. So let's start from evidence from animal models. So the model I'm going to present is a very interesting one. It's a rat model of maternal care in early life. So certainly there are different ways we can measure maternal care in humans, but in rats, one way investigators do that is to get the rats in a chamber and to measure how often the dam, the mother rat, licks and grooms their pups. And so we have, you can imagine that there are really scores of postdocs and students who spend their time counting or quantifying the amount of licking and grooming that a mother gives to her pups. And you can see that there is quite a variation, and this variation is mostly stochastic. I mean, there is no particular reason why these rats are different, but clearly there are rats that are really not doing that well, and rats that seem to be hyperactive. They really give a lot of attention to their pups. And what is really interesting is that it has been shown that the amount of licking and grooming affects the programming, not just the functioning, but the programming of the offspring's stress response system. In particular, I would like to review for a second the HPA, hypothalamic pituitary adrenal axis, because it's particularly important for this experiment. This is an axis where the brain, through the hypothalamus, the pituitary, and the adrenal gland produces cortisol, which is a stress hormone. And this is tightly regulated. We have a diurnal cycle where cortisol is up in the morning and goes down in the evening. And also, we are always ready to pump lots of cortisol in our system should we need it in case of stress. And of course, there is a feedback of cortisol. When there is enough cortisol into the circulation, the cortisol level is read back by a specific parts of the brain. And when there is enough cortisol, the brain tells in the circulation, the brain tells the hypothalamus and the adrenal to stop producing cortisol because there is enough concentrations. So it's not just, I mean, this would be amazing. For instance, for me, I'm pretty passionate about cooking. It would be really amazing to have a sensing system like that when I cook, something that I don't have to measure too much, but something that tells me exactly once I dumped enough salt in my preparation, something that tells me that it's salted enough. And indeed, this is what you do when you taste it. I mean, you try to taste the preparation and see whether there is enough salt. So the brain does exactly that. Not only puts the right amount of cortisol in the circulation, but also has sensors that sense when the concentration gets to be high enough. So going back to our LG licking and grooming model, what our colleagues in Canada in Dr. Mini's lab a few years ago found out is that the offspring of mothers who don't lick and groom their pups very much tend to have higher responses to stress. And this is shown, for instance, by the amount of corticosterone. Corticosterone is the rat equivalent of cortisol. Rats don't have cortisol. Humans do have cortisol. So you see here that corticosterone was pretty higher after a stressor was administered in the rats with low licking and grooming. And what is really important is that this continues to happen as the pups become adults. So this is something that happens in the first few hours, days of life, but it's really a program that is happening. The brain gets wired differently. And for all their lives, these rats are more fearful. They tend to get stressed much more and much earlier. And this seems to be also due to higher corticosterone. The reason why this is happening, though, it's because the brain of these rats is much less sensitive to the circulating corticosterone. So they have higher glucocorticoid receptors. Glucocorticoid is a name for all the entire class of cortisol-like substances. So both corticosterone and cortisol are glucocorticoids. So there is increased glucocorticoid receptors in the, so this is actually the other way around. So the mothers who lick and groom a lot have pups with higher glucocorticoid receptors in the hippocampus, which is the region that is particularly important to shape emotions and also to control the secretion of cortisol. And so this is particularly important because then what we have is that the low licking and grooming mothers have rats which pups with much less levels that tend to be insensitive to the levels of cortisol. So it's almost like you are cooking and you can't taste the salt and you keep dumping salt because you can't feel it, and your preparation becomes very salty. This is what happens by not sensing the cortisol. This rat becomes very stressed and very highly responsive to stressors. But what is really important for us is that this is not just happening at random, but it is programmed by DNA methylation. So the rats with high licking and grooming happen to have very low methylation in the glucocorticoid receptor. And so their glucocorticoid receptor in the hippocampus is highly expressed, and vice versa, the low licking and grooming have higher methylation that silences and stops the production of the glucocorticoid receptor in the hippocampus of those rats. And this is how the programs exist. So methylation programs those genes and those brains to be that for an entire lifetime. It is particularly important thinking about the effect of early stressors on human life. The investigators also asked themselves whether this is really due to the licking and grooming experience, or maybe a little earlier, and to assess that, they decided to do cross-fostering experiments. Cross-fostering is a very simple experiment where you get rats from moms who are known to be high LG and moms who are known to be low LG, and you swap the pups. So you can see that these are this experiment, this part is exactly like this one. These are moms who are dams who are low leaking and groomer, and the pups were assigned to them, were born from them and then assigned to them. So they were not swapped. Here they were not swapped, it was high and high. And here instead there is the real cross fostering. So the high pups born to high LG that are swapped and raised by low LG and vice versa. And you can see that there is a little bit that changes, but most of the difference remains, which suggests that most of the effect here is really due to the behavior of the mom in the early life of the pups. And so this is really a summary, and just to make sure everyone followed, I'll go through the summary. So what happens with lower pup licking and grooming? So by pup, they were licked very little by rat mothers. They had higher methylation of this receptor, the glucocorticoid receptor, GCR or GR, in the hippocampus, which was reversed with cross-fostering, persistent into adulthood. And these rats, early on and later in adulthood, were more fearful. They had higher stress responses. And the pups, but what is really interesting is that this doesn't seem to stop, because pups raised by low LG mothers became themselves, if they were females, low LG mothers. So it seems this also seems to create a cycle of behaviors that continues through generations. And this is particularly important thinking about humans, thinking about cycle of stress, thinking about why some of us are more stressful or stressed out or easily stressed out than others, and so on. And so after setting the stage with all these experiments, I would like to show how things might work in humans. And at some point, Alexandra Shields from Harvard, Julie Palmer from Boston University, and I got together, and we were all very fascinated by the studies on rats that I just described. So we got together and said, can we study the same mechanisms in humans? And Julie Palmer in particular had been working for many years on this study, the Black Women Health Study, and we set forth to try to understand whether we could, in a way, translate the study, the rat study I just showed, into this study, mostly to assess whether a history of child abuse in early life was associated with methylation levels in the HPA axis. And I'm going to show particularly the results we have on the glucocorticoid receptor, which is the same receptor as in the rat study. And this study was started by NCI in the United States. Participants were enrolled in 1995. We had questionnaires. It was a large study of women back then, 21 to 69. And they were followed up every 2 years by mail and questionnaire. And early on, they were able to get a blood sample, which we used to isolate blood DNA, and that we used to do DNA methylation studies. And please note that while our colleagues working on rats use the hippocampus, we use blood. So it's particularly interesting to think whether blood is a good tissue, because of course it has completely different functions than the brain. So there were the blood was collected only on 500 participants in this study, and this is what we used for our study. And we selected particularly 295 women out of these 500 with lots of data on psychosocial measures. And the outcome was percent methylation of the glucocorticoid receptor, particularly a gene, an exon in the glucocorticoid receptor that seems particularly functional. And this is what we found. So if women reported a history of child abuse, and you can see the frequency of physical abuse during childhood and the severity of physical abuse during childhood, there was an increasing level of methylation in the glucocorticoid receptor in blood. So please note that they are reporting abuse when they were children, but we are measuring their blood in people who are now middle-aged. And so this really shows the idea of programming or long-term changes in DNA methylation. And so this really is interesting because it seems to parallel very well the study on rats, and potentially what happens in rats happens also in humans. I would like to show quickly also another study, which is completely different. These are workers who were shift workers and night workers in particular. And we show it's a completely different type of stressor. And the study here is cross-sectional. These people were workers at the time, night workers at the time of the study. But what we found is that the number of years these people had been on night shifts was also associated with a level of glucocorticoid receptor methylation in their blood. And it is really particularly interesting because it seems that this type of glucocorticoid receptor tracks along with different types of stressors or stressful conditions. So at this point, I would like to spend 10 minutes or so to tell you about transgenerational inheritance. And and I'll take a sip of water just to pause. So I'll start from back in the 1700s or early 1800s. So you might remember that at some point in your education, you might have heard of two conflicting theories about evolution that were popular 200, 300 years ago, 200 years ago. And one championed by Charles Darwin, that is the one that really we're familiar with, and one championed by a French scientist by the name of Jean-Baptiste Lamarck. And I mean, Lamarck, I don't know how you felt about this, but Darwin made a lot of sense after being educated to it. But Lamarck seemed a little out there, a little peculiar. And he had ideas that were and examples that were a little weird. And it was the idea that one example, for instance, the example of the blacksmith. He had noticed that families of blacksmiths tend to be bulky, tend to be very muscular. And a blacksmith is muscular, and his sons, and please, sons comes from him, comes from Lamarck, are also pretty much built up. And this idea is that this is, in a way, inherited biologically. Through the sperm, there is some type of signal that signals the fact that a blacksmith has worked out all of their life, had bulked up quite a bit because of his job, and now this type of information of being bulked up get transmitted to his sons and perhaps to his daughters, but he really thinks about the sons, which is interesting, and I'll show you in a second why. The other example was the example of the giraffe, a giraffe living the idea of the giraffe and the long neck is that the long neck was an evolutionary trait that the giraffe acquired by stretching their neck little by little because of the high height of vegetation in certain areas to reach the vegetation for food. And this stretching was transmitted to the multiple generation. So this is particularly interesting because thinking back at the example of the stressors of the rat model and the human studies I presented, if Lamarck is right, what we do to our bodies today, whether we take care of our body, whether we smoke, we drink, whether we get ourselves into a life that is too stressful, whether we take care of slowing down, may not only influence us, but may influence also the children we might have in the future. And not just because of behavior, because of course our behaviors and our fostering our children really matter, but because of signals that get transmitted through the gametes, through our germlines, through the sperm or the oocyte. And so, I mean, Lamarck has been dismissed for so many years, and you really feel bad for him. But now Lamarck is coming back, and I'll show you how. For instance, a few years ago, Diaz and Ressler published this study in Nature Neuroscience, where they showed that if you fear condition a rat to a certain odor, they used an odor that smells like cherry blossom, very neutral, so nothing that a rat would have any reason to get worried. But I mean, fear condition is a very simple experiment that's been around for centuries, where you give the rat a cue, like a cherry blossom smell, and then you give them a very unpleasant, different experience, particularly in this case, an electric shock. So after a while, the rat is very smart, and anytime there is cherry blossom in the air, anytime they can smell cherry blossom, they start to be very fearful. They start to show a very fearful behavior because they know that the electric shock is coming. And so this is just a simple fear conditioning experiment. They did it on male rats, and then they mated the male rats with females, and they got down to multiple generations. And what they showed is that when the first and the second generation were exposed to the cue, to the cherry blossom, even without being trained to the fact that the cherry blossom is bad, they started to be startled. They started to have this fear reaction that is very typical and visible. And this really seemed to indicate that the experience of the father was transmitted down to the sons. And please note that for some reason, this was observed only in male rats. So perhaps Lamarck was really, really right that this goes through the sons and also to the grandchildren, or better, the grandsons. And it's particularly important that you can see that on the grandsons, because, you know, when these rats are shocked, the sperm is in here. And by the way, they found changes in DNA methylation in the olfactory genes in sperm that might reprogram perhaps the odor, the olfactory senses of these rats. So, I mean, when the rats are shocked, the sperm is there. So you might think that the sperm gets immediately reprogrammed, and this is why these rats are fearful. But, you know, the second generation has never been even remotely exposed to the shock. The sperm that make these rats didn't exist here when the shock was made. So potentially this could go on forever. And I had to point out that in terms of evolution, this would be particularly important because, you know, as humans, we don't do much with our smell. We really use it very little. If you have a dog, you can understand how their smell is much more powerful. And for rats is particularly important because rats are little things, and the biggest enemies, the biggest threats to rats are predators. And there is only so much you can do with your hearing, only so much you can do if you're a rat with your vision. There is a lot more you can do with your smell. If you can smell a danger, a predator, you are in good shape. So you can imagine how this small rat would have all the interest of letting the future generation know that there is a new predator with a certain smell, there is a huge threat, and you need to pay attention. And this is apparently what happens. I have to add that because the authors were challenged to think that perhaps there was some behavioral transmission to the rats, perhaps even some communication. I don't really speak rat language, but you can think that the that the male may speak to the female and tell about all the dangers that are in the world, and they can speak, the female can speak to the offspring. So they also did the same experiment with in vitro fertilization. So they didn't had the rat's mate, but they just got the sperm and inseminated, actually did in vitro fertilization and then implanted the embryo into the female. And that really broke any type of communication between the rats, between the male rats and all the other rats are down here. The issue is that, so this seems to show that we are in a new brave world where there is this thing where things that we experience in our lifetime can be transmitted biologically to our children. There is so much we transmit through behavior. And the question is whether this can be translated to human studies. So there is some evidence that this might happen. For instance, our colleague Andrea Roberts, we collaborated with her on this project, showed that men with early life childhood abuse, when they become adults, they have differences in their sperm in DNA methylation. So this seems to be exactly the parallel of the rat study I just showed. It seems that those of us who had a history of child abuse have changes in DNA methylation in our sperm, and the question remains whether these changes in DNA methylation matter for our children. But this is really, in a way, unsettling and important to be followed up. Also, Andres Cardenas showed that looking at the mothers, prenatal maternal antidepressants changes the DNA methylation of the offspring. So also this clearly can be a pharmacological effect or can be a marker of having depression, and this changes the offspring DNA methylation, and this on a specific gene, the zinc finger protein 575 gene. And this was found in a human cohort in Boston and replicated in a human cohort in the Netherlands. So in summary, I would like to wrap it up. I know I covered a lot of ground, so I will be happy to hear any questions. Epigenetics, in particular DNA methylation, can provide molecular traces of past exposures to stress, and very important for programming. Of course, transgenerational program effects are particularly important, are the most intriguing part of what I spoke about today. And the question is, are there transgenerational cycles of exposures, stressors, disparities? And unfortunately, the examples I have to say are still sparse, less sparse in animals, definitely very hard to study in humans. And you saw how the examples I gave you were very short. Measures of sperm, not of multiple generations, measures in the offspring, not in adults even, not in multiple generations. But down the line, I think what we all want to do is to think how and whether we can keep our epigenome healthy. And there is a lot of opportunity for research leading to primordial, primary, and secondary interventions, and perhaps even tertiary. So this is all I had to tell you, and I hope that you enjoyed my talk. I would like also to tell you, for those of you who are interested in more epigenetics, that we run every year here at Columbia, and most likely this year is going to be online, a 2-day intensive training that we will be offering in June 2022. And we are also thinking about a special session in January 2022. So it might come up soon if you want to learn how to do this, and also to learn how to do data analysis on large epigenetics, particularly epigenome-wide data. You may want to learn more about this, and this is part of our SHARP training at Columbia. And this is part also of a much larger offering. We have 20 of these boot camps ranging from data science to NIH grant writing to omics. So there is much more, and you can look it up online. So thank you so much. I hope I left enough time for questions, and happy to be here again.
Hello, thank you, Dr. Baccarelli. There are currently no questions in person. If there's any other questions, please write them in the chat.
Well, if I can just comment, not really a question, but a comment. I think what Professor Baccarelli has explained to us is that the importance of epigenetics. In epigenetics, there's no change in the genome, but environmental factors could actually influence the way the gene is being expressed. And so as we look at genetics and suicide, and certain environmental factors, for example, trauma, abuse in childhood, what impact that could have on suicide and psychopathologies later on in life. And I think this is an important aspect for us, even as we contemplate the establishment of GRIT and the kinds of research that we can do to understand in our environment here in Guyana, what are the mitigating factors that might have contributed to suicide? And maybe we need to look at childhood, at some of these factors, particularly in children, and maybe follow them all through, look at family studies and so that we can have understanding, so we can have our intervention. I just wish to make those comments, and I wish if Dr. Baccarelli can also comment on that.
Absolutely. I had to add that if I can add a little bit of personal history, I started my training as a geneticist. I was really interested in genetics. And at some point, I started to get interested in epigenetics because the epigenome is modifiable. I mean, while the genome is very hard to change, I mean, now we are doing epigenetic editing, and the idea of modifying some sequences of the DNA is a reality, though certainly this can be done only at the moment, at least for single gene disease, which unfortunately are only a very small minority of diseases. But the epigenome is changeable. That really means that you can detect the influence of the environment, but potentially you can find ways to put it back to where it's supposed to be. I had the idea of the healthy epigenome, that, I mean, you can design interventions to modify the epigenome and to reshape it, to make to wipe away the negative effects of early life experiences or of stressors that might, for instance, lead to suicide. And I mean, in a way, this is also sci-fi. But for instance, something I didn't show on the Black women health studies, that we had that effect on of early life abuse on the glucocorticoid receptor methylation, but the effect, the negative effect, was attenuated in those women who had higher levels of social support, which might suggest that through early life experience, this terrible early life experience create a negative program on the epigenome, but there are buffers in later life that can help attenuate the effect. And actually, those with high level of support had no effect whatsoever. So in a way, it's really interesting for the epigenome, because our destiny is not written in there. It's not written in the DNA. It's not written in the epigenome. It can be modified by appropriate interventions. And I hope there is also room for collaborations with this group, thinking about this type of ideas.
Good morning. I'm sorry if I initially stated no questions, but I had one after thinking about the study or the experiment with the rats and the passing down of the genetic information from male and interestingly to other males. Has there been any other similar with females to see how that trend goes? Over.
Yes, there is quite a bit that happens with females. They're much harder to study for a very simple reason. And actually, I think I had a slide here. Maybe I can show. Yeah, actually, I can show this slide if you don't mind to explain what is the difficulty here. And this is important to know. Yeah, if you can see, I mean, this mostly what was done in the males. These are humans, but you can think about the rat experiment. If you study the males, I mean, the first generation here is already. I mean, of course, this person is already, if it is affected, if they are affected, it might be due by the fact that the reproductive cells were in the body of the father that is impacted by changes, for instance, in stress. And then you can study, and this is what we usually call intergenerational effects. So it's not yet transgenerational. Intergenerational because the cells that will make this person are already here when the father is exposed to stress. And you can see real transgenerational effects down here when you have in the grandchildren, the grandsons. In females, the situation is different because you can have stress to the female, and the stress of the female can change the female body, so it can directly expose the fetus. The fetus has, of course, all the fetal cells, but also the reproductive cells that will make the second generation. So if you study the female lineage, you are still looking at intergenerational effects in the children and in the daughters and the granddaughters. In order to see transgenerational effects, you need to come down here at the F3. And this is particularly important because, going back to your question, there is quite a lot that has been done, but many of the effects seen in animals, and this is really very difficult to study in humans, really seem to stop at F2. They seem to stop at the granddaughters. There is quite a few, though, that continue down to the great granddaughters. So there is a lot ongoing. I think in part because of this more challenging experiment, lots of animal labs like to study the male. And this perhaps selfish, perhaps just getting out of trouble, perhaps making sure that the student who work on the experiments actually graduates and doesn't have to be 10 years there to this multiple generation study. It's really an issue of cost as well. But going to your question, there is much more research on males than females and much more success in males than females. And perhaps also it's much harder to keep the conditions pristine for multiple generation, but there is evidence in males. And certainly there is evidence, if not of transgenerational effects, of intergenerational effects, which are shorter across 1 or 2 generations. I hope that answers your question.
Yes, and I'm assuming in terms of a comment, I'm assuming because it seems like it's from male to male and female to female in terms of the study. I'm assuming going across, for example, all in the next generation, both males and females. I'm assuming that the issue to study those would be the same issues that would have been for the female to female in terms of the difficulty.
Yep. Absolutely, absolutely. And I have to say that the experiment I showed found adverse influences only in sons, in males, but there are studies that show that effects that are found both in males and females in the subsequent generations. So it seems to be pretty specific on the type of model and the type of gene that is being studied. Most likely it's the gene that determined that, but there are certainly some genes that might have transgenerational or at least intergenerational effects, both in male and female offspring. And it's complex, but we need to keep in mind that it might be gender specific. but it might not be.
Yes, noted. As I assume that it would be interesting also to study why it is gender specific in terms of activation and inactivation across the genders. If there's anything specific in the female gender that suppresses or, as you stated, silences that expression as opposed to in the male.
Absolutely, would be a lot, absolutely interesting. Fantastic. It looks like we are out of time. I don't want to call it too early, but if there are any other questions, happy to correspond by email. I'm always available. And thank you so much for having me.
Thank you very much, Dr. Andrea Baccarelli, for sharing your information. I'm certain it will be useful to us in our own research and our own work.
Thank you so much. Great pleasure. Happy to be a resource at any time.